There we go. Okay, there's one last funny little wrinkle that I wanted to mention before we move on. We talked about the basic Hardy-Weinberg model. I've talked about how you incorporate selection into that model. We've looked at inbreeding and how we incorporate that. There is additional math that you can do to model the effects of migration in and out. You played with that in the computer simulation that we did, although I didn't really introduce the equations for it. There are ways we can model the effects of mutation. I didn't get involved with that. Mutation is important because that's ultimately where alleles come from in the first place. But by itself, realistic mutation rates are slow enough that by themselves, they don't drive a population off of Hardy-Weinberg equilibrium, not in realistic periods of time. There's a couple of other wrinkles that we'll add later. The last wrinkle I want to throw at you is something called density-dependent selection. And that's cases where the fitness that we assign to genotypes is not fixed. Now, in the models that we've done, you set your fitness in the computer program, you set your W's, and then those W's stayed the same throughout the whole simulation. You could run that for 10,000 generations and the fitness of A1 would always be 0.7 and the fitness of A1, A2 would always be 0.9 or whatever it is that you set it to. One of the things that we find is that the fitness of a genotype is not always constant. Sometimes fitness can be a function of the frequency of the genotype. Sometimes you can have particular genotypes that are selected for because they are more rare or because they are more common. And there's a really good example of this from Parasotus eccentricus. It's kind of hard to see in these pictures, but Parasotus is native to Lake Tanganyika in East Africa. And it has a rather odd feeding strategy. It's called lepidophagy, which refers to fish that eat the scales of other fish. This is a fish that will sneak up on another fish and take a mouthful of scales and swim off. This can be bad for the fish that gets bitten because infections can get in if there's a big wound. But the scales do eventually grow back. So this is not really a parasite, it's more of a micropredator. If you were in parasitology, you might remember that term. Anyway, Parasotus is weird because in order to attack efficiently, it's hard to sneak up on a fish because they've got bulgy eyes that are on the sides of their heads, which means they've got an extremely wide field of view. So Parasotus has to approach from behind, and to attack more efficiently in that direction, its mouth is twisted either to the left or to the right so that it can approach from behind and to the side and grab a quick mouthful before the other fish is aware of it. So Parasotus eccentricus comes in either left-handed twist or right-handed twist models. Up at the top, those top two images are of a fish with a mouth twisted to the right. At the bottom, I think that's a fish with a mouth twisted to the left. And somebody measured the frequency of the different variants of Parasotus from season to season. That's a graph of the frequency of lefties over a decade. And what I want you to see is that it zigzags. It oscillates fairly regularly, looks like with a period of roughly every 4 years. lefties become more common, and then they diminish in frequency, and the righties dominate, and then the lefties become more common, and so on. Think about this. You know, if you're a scale-eating fish and you're eating the scales of other fish, those fish can learn, and those fish are going to try to avoid you. So the strategy that works as to which one is better, being a lefty or a righty, the answer is neither. The answer is whatever your enemy is not expecting. Right? So if the right-handed fish, the ones with their mouths twisted to the right, are the ones that are most common, the prey is going to be aware of that. They're going to be expecting an attack from that side. If you're a left-handed fish, right, you're attacking from the side that they are not expecting, right? You're attacking from an unfamiliar direction. So when the righties are common, the lefties have an advantage. If the lefties have an advantage, they're going to eat more, they'll get more food, they'll breed more, and there will be more lefties. But when the lefties become common, the lefties lose the advantage, right? Because their prey is expecting lefties now. And that means that the righties have an advantage because the righties are now able to attack from a direction that the prey is not expecting. And then the righties become more common. But when they're more common and the lefties are more rare, selection favors the lefties. I take it you see how this plays out. This selection doesn't favor either having a left twisted mouth or a right twisted mouth. Selection favors whichever strategy is less common. And then when that strategy becomes more common, it's no longer favored, and the other strategy is favored. And so you get this fairly regular oscillation in phenotypes, but neither one ever becomes dominant. That's density-dependent selection. Another example turned up in 1997. There's two behavioral types of fruit fly larvae. And you can test this by putting fruit fly maggots on a petri dish. Rover larvae tend to crawl for very long distances when they're feeding. Setter larvae tend to stay in one place as they feed. They tend not to move. So the rovers will make long trails and the setters will just kind of stay where they are. It turns out there's a genetic basis to that. And neither one is permanently favored in the population, but what keeps both alleles in the population seems to be density-dependent selection. Right, if you're a setter, there's an advantage to that in that you're not spending a great deal of metabolic energy traveling, right? You're just staying right where you are and eating, and you're not burning a lot of calories, so you grow faster. The problem is when there's lots of setters in one place, they tend to deplete the food supply pretty quickly. So when setters dominate, rovers are favored because the rovers travel away and they can find new sources of food away from the ones that the setters have consumed. So when rovers are rare, rovers are favored because they don't exhaust the food supply. When rovers are dominant, setters are favored because being a rover also has some risks. You burn more energy traveling to find new food sources, and there's also a chance that you won't find them and starve. So neither one of these traits is strongly favored all the time. Selection favors whichever one is most rare. And as each one becomes more common, it loses its advantage and the other one becomes favored. And so density-dependent selection maintains both alleles in the population. Neither one is ever driven to zero. So that's density-dependent selection for you. Okay. I will come back to a few things that we've skipped, but what I want to do is move on now. I am going to introduce some math, but this is going to work out a little differently. I want to look at selection on continuous traits. We've talked about, I don't know, lupins that are either blue or pink. In your genetics classes, you probably started with Mendel's laws, which were worked out on peas that could be either purple flowered or white flowered. Or, you worked on, peas that could either be yellow or green, smooth or wrinkled, fruit flies with either red eyes or white eyes. We've done all of our work so far on, situations where there's just one gene with a few alleles governing a well-defined trait. The problem is that an awful lot of traits that we're interested in don't follow those rules so neatly. Everything from in humans, human height, human build, shape of human features, even human intelligence. They've all got a genetic basis to them, and they're all things that selection at least potentially can affect, but they're not down to single loci. As I trust I made perfectly clear, we are not a species where feeble-mindedness is governed by one single gene with two alleles, one dominant and one recessive. We are infinitely more complex than that. And in fact, so are most species with most traits. So I was in grad school when this book came out, a book by Richard Herrnstein and Charles Murray. Murray being a psychologist at Harvard and Herrnstein a political scientist at a group called the American Enterprise Institute. It's called The Bell Curve. And you'll notice it says that this was the controversial New York Times bestseller. It definitely was, because the Bell Curve made arguments like this. Ethnic differences in cognitive ability are neither surprising nor in doubt. Large human populations differ in many ways, both cultural and biological. It is not surprising that they might differ at least slightly in their cognitive characteristics. One message of this chapter is that such differences are real and have consequences. So what he's talking about is, first of all, that intelligence is genetically inherited. And second, just because people of different races inherit different physical traits, they've also inherited different mental traits, and there are differences in intellectual ability between different racial groups in America. Boom. I think you can see why this rather bothered people. Unfortunately, it also didn't bother certain people. This is one of the illustrations from that book. What they took is the NLSY is, I think, the National Long-Term Survey of Youth. What you're looking at is the distribution of American IQ. Distributions for IQ and for a lot of traits follow bell curves, and this is typical for a feature that has lots of contributing factors. If you were to roll 10 dice and add up the numbers that you got on the dice, record that number and do it again and again and again, you'd get a bell curve. In that it would be, if you were rolling 10 dice, it would be extremely rare that you'd get 10 ones, right? So very rarely would you get a total number of 10. And it would be extremely rare that you'd get 10 sixes. So 10 would be very rare, 60 would be very rare, but something like 35 would be much more common because there are many more ways you could throw those dice and get 35. So if you plotted your numbers after 10,000 dice rolls in a bar graph or something like that, you'd end up with a bell curve. And intelligence also tends to plot out on bell curves like a lot of traits. You know, there are some people that are absolutely brilliant and some people that are cognitively impaired, and most people fall somewhere in the middle. No different from, let's say, height. You know, there are some people that are NBA centers and some people that are Kentucky Derby jockeys, and most of us are somewhere in the middle. And IQ follow that bell curve as well. What Murray and Herrnstein claimed is that you can see there's a definite difference between IQ in the European American population, where it looks like the peak of the bell curve is at about 105, and the African American population, where the peak of the bell curve is about 80. In other words, the African American population averages about 1 standard deviation less intelligent than the European American population. Notice you've still got some smart Black people. You know, I've got Black friends. Sorry, that was being sarcastic. You still have some intelligent Black people and you still have some stupid white people, but the average is less. That distribution is left shifted for African Americans compared to Americans of European descent. And the book is a long argument about this difference being real and this difference also being innate, being something that is in the genes, something that we really can't do very much about and probably shouldn't try. Because if the African American population is genetically doomed to being less intelligent than their white kin, then clearly no amount of affirmative action and preschool education and all of that stuff is going to make any difference if it's just programmed into the genes. This stirred up quite a bit of controversy in some circles. For the record, I think the book is utterly wrong, and I certainly don't support their conclusions. A lot of people agreed with me. This book that came out, The Bell Curve Wars, included, there's a very good essay in there by Stephen Jay Gould, an evolutionary biologist at Harvard. I think he died about 10 years ago or so. But his essays on evolutionary biology are mostly very much worth reading, although towards the end of his career, he did tend to start rambling quite a lot. But, you know, this touched off response. This touched off considerable debate. And I want to take you into some of that debate. I think I'll be able to show by the time we're done exactly where the bell curve went wrong. And it's important in this day and age that people in general understand why the bell curve went wrong, because the arguments that it puts out are anything but dead. But I want to delve a little bit deeper into this, and we'll come back to the bell curve, but I want to talk about how we actually do model the genetics of quantitative traits and how we can apply selection to them. Okay, so we've already seen this. Quantitative traits do not have a small number of phenotypic states. They vary continuously. Height, weight, length, body part size, you know, size of your fingers, size of your ears, size of anything else, intelligence, susceptibility to disease, things like that. They're all continuous variables, and they're determined by multiple genes. They also are frequently determined by the environment. Your height is partly a function of the genes that you inherited, but it's also a function of things like childhood nutrition. People who have been starved as children often grow up to be shorter than people that were not. People that eat light diets weigh less than people that slam down 10 quarter pounders every day. Right? Your weight also has a genetic influence to it, but there's also an environmental effect to it as well. So do a lot of things. And one of the things that we're going to have to try to disentangle is the effects of genes versus the effect of environment on phenotype. We can model how selection works on quantitative traits in three basic ways, although you can find more complex examples in nature, but we can break them down to directional selection, stabilizing selection, and disruptive selection. In directional selection, you're basically taking a bell curve and you're shifting it in a particular direction. You're moving the mean phenotype. By the way, if a trait truly is represented by a bell curve, if it truly fits what's called normal distribution, then the peak of that bell curve is also the mean, and it's also the median. If anybody has forgotten, if you have a set of data, the mean is the average, the median is the midpoint of the distribution, halfway between the extremes, and the mode is the value that appears most frequently. And if you have a normal distribution, the mean, median, and mode are the same thing. The average value is also exactly in between the extremes, and it's also the most common value. Okay, so if you have selection that shifts the mean, it's directional selection. Sometimes you get selection that favors the mean phenotype and acts against the extremes, and we call that stabilizing selection. And sometimes we get selection that favors the extremes and acts against the mean, and we call that disruptive selection. Let's look at some examples. Okay. Peter and Rosemary Grant have been going to the Galapagos for something like the past 50 years. I believe they're based at Princeton University, but they've been spending field seasons there every year, and they've been following finch populations in very close detail. And they've looked here at a population of Geospiza fortis, the large-beaked ground finch. I believe this is on only one of the islands that used to be called Daphne Major. Now, what they did was capture the birds gently and measure beak size and measure several different dimensions of that beak. And then there's a way called, I believe this is principal component analysis. You can take multiple measurements of something and boil them down into a single number. So you could take one beak and measure length of the upper part, width of the upper part, height of the upper, length of the lower, width of the lower, height of the lower, maybe some more things. So that gives you 6 numbers, and you can't plot all 6 numbers at once because it's hard to find 6-dimensional graph paper these days. But what you can do is use a mathematical technique called principal component analysis to take those 6 numbers and boil them down into a single one that represents most of the variation in your sample. That's what's graphed here on the y-axis. It's labeled overall size. And if you're wondering how a bird can have a beak size of -2, that size is not really one single measurement. That size is based on a whole bunch of measurements. And it's just a number that expresses how different the beaks are from each other. So in 1975, you can see is when this begins. Males have bigger beaks, females have slightly smaller beaks. Beak size stays pretty close to the same until mid-1977 when there's a drought. The Galapagos get hit with devastating droughts every few years, and this is connected to the El Nino cycle, of which I'm sure you've heard. And what I want you to see is that the average beak size increased tremendously during the drought. I mean, a lot of birds died. You're not really seeing that in this data, but the ones that died were the ones with smaller beaks, because the birds with smaller beaks didn't have large enough beaks to crack big, tough seeds. Birds with the larger beaks had more food because they could crack larger and tougher seeds. And over the course of less than a year, that's what we see. You know, what those dots on the graph are is the mean of overall size, and that average shifts. Goes from about 0.5 to 1 in the case of the male birds, goes from about negative 0.2 to positive 0.3 in the case of the female birds. Those lines, those vertical lines that you're seeing, that's probably standard deviation, right? But the dots that are connected by the colored lines, those are means, that's average beak size, and the mean shifts because of selection. So that's a very nice case of directional selection. Here's a human example of stabilizing selection. This is a little bit weird to read because what you've got is a bar graph and a dot plot superimposed on each other. The bar graph, right, the thing that looks like a funky skyscraper, is birth weight graphed against percentage of the population. So about 20% of the human population, at least at one particular hospital, I think this was in Britain, 20% of the population of newborn babies was about 7 pounds. And then about 15% of the population is at about 6 pounds, and another 15% was at 8 pounds, and so on. You can see that very few babies are born at 1 and 2 pounds. Very few babies are born at 10 and 11 pounds, because ouch. Most babies are born pretty close to the middle of that distribution at about 7 pounds. The mean birth weight is just a little above 7. You can see it marked down there below. The graph shows you of dots whose y-axis is on the right side, shows percent mortality. Babies with low birth weight are more likely to die. I think this data is a few decades old. This would still be true today, that if you're born extremely small, you're probably going to go into, neonatal intensive care. This data may actually be from the 50s when, NICUs were not nearly as well developed as they are now. And if you were born prematurely, you were quite likely to die. My parents' first kid, that happened to. He was born 2 months prematurely. If it happened today, they probably would have saved him, but in 1968, they couldn't. So it goes. You also have an increase in mortality for very large babies because labor takes longer. They're probably more likely to be damaged passing through the birth canal. There may be other complications I'm not aware of, but very large babies have a higher risk of dying. Very small babies have a higher risk of dying. The smallest risk of mortality is in the middle. And in fact, that optimum birth weight right there, right, that's the low point of that curve. That's the birth weight that has the least chance of dying, is very close to the mean birth weight. The majority of babies are born very close to the optimum birth weight. Birth weight does seem to be partially heritable. So if you're a very large baby, you have an increased chance of dying and you won't pass your genes on. If you're a small baby, you have an increased chance of dying and you won't pass your genes on. If you're in the middle, you've got the greatest chance of survival. And whatever genes you've got that might influence birth weight, you have the greatest probability of passing those on. So that's stabilizing selection, stabilizing that favors the mean and does not favor either extreme. Disruptive selection is harder to find, but I found a case study of it in a study of an African bird called the black-bellied seed cracker. Very attractive bird right there. This is a bird where the population does suffer pretty high mortality every year. And Bates Smith, back in 1993, took a bird population, measured the width and length of the lower mandible, right, the lower jaw, at the beginning of the season and then at the end of the season. His measurements at the beginning of the season are shown in the bar graphs on the left in light color. And then in the shaded bars are the survivors. So in particular, look at that lower bar graph. It's not perfectly a bell curve, but you can see that there were few birds with a very short mandible, few birds with a very long mandible, and most are kind of in the middle. But the survivors, with those gray bars down here, tended to be the ones with either fairly small mandibles or the ones with the largest mandibles, not the ones in between. I'd have to go back and check the paper to figure out exactly why this is happening. It may be a situation where in their environment, there are small seeds and there are large seeds, but there aren't any seeds in the middle. If that's the case, this is a case of disruptive selection where there's selection against the mean and selection that favors both of the extremes. Okay. One of the fun things that competition can do is create something called character displacement. You've got three heads of three different Galapagos finch species: small-beaked, medium-beaked, and large-beaked. If you look at the bar graphs below, you'll notice that there is only the small-beaked groundfish, finch, on the island of Los Hermanos. And on the small island of Daphne, there is only the medium-beaked ground finch. And on the islands where the birds don't compete, the phenotypes are very different. On the islands where the two don't compete, the phenotypes are not very different. They overlap quite a bit. On the other hand, if you go to Floriana and San Cristobal, you'll notice that they are more different from each other. The Geospiza fuliginosa graph is shoved over to the left, and the medium-beaked ground finch, I think that's G. fortis. I can't read the name very well because I've got a little zoom bar up on top of it. But the medium-beaked ground finch has bigger beaks on average, and the small-beaked ground finch has smaller beaks on average than they do when they're in populations where they live alone. That's what we call character displacement. What's presumably going on is that selection is favoring birds that aren't competing with each other. So Geospiza fuliginosa it's advantageous if they're competing with the medium finch. It is advantageous for them to specialize on smaller seeds that Fuliginosa can't get. And the medium-beaked ground finch is the same. If they're competing with the small-beaked ground finch, selection favors larger beaks so that they're not competing with Geospiza fuliginosa. And that's character displacement. Competition tends to reinforce species differences. It's happened again on Pinta and Marchena, where Geospiza, the medium-beaked ground finch, has to compete with the large-beaked ground finch. And so you can see that that bell curve of beak size has become narrower. Okay, so that's character displacement, and that's a wrinkle that can happen in selection acting on continuous traits. I'm going to take you through a bit of the math that we use to model this. When we have some quantitative trait, like we could measure everybody's height in this class. I mean, I couldn't do it now because we're scattered, but I could have you report all of your heights. And that would be our data set. And of course, we could take the average and we could calculate something called the variance. And that's the mean of the squared differences between each data point and the average. So I could calculate this class's average height. I could calculate the difference between each individual's height and the average. You know, one of you might be 2 inches taller and one of you might be 3 inches shorter and one of you might be 4 inches taller, So I can take all of those differences, square them, and then take the mean of those and I'd have the variance. You can intuitively think of variance as a measure of how wide that bell curve is. If I took the square root of the variance, I'd have the standard deviation, by the way. I know you've heard of that. And if we have a population that has some phenotype, we can measure the phenotype of the whole population and calculate the total phenotypic variance and call that V sub P. So I could measure the height of all of you, calculate the mean, use that to calculate the variance. And intuitively, you can think of the variance as being an estimate of how narrow or how wide that bell curve looks. The fun thing comes when we try to make like MC Hammer and break it down. Some of what's making, okay, let's go back to thinking of the class and measuring our height. We've all got different heights. I have yet to meet you in person yet, but some of you are pretty tall and some of you are pretty short and some of you are kind of in the middle. So we could calculate the variance for the class population of height, or the variance in height for our class population. Some of that variance comes from genetic variation in the population. Some of that variance comes from environmental variation in the population. And we can just write that your total phenotypic variance is the variance due to genes plus the variance due to environment. Some of you might be tall because you've got tall parents. Or no, I'm not putting that right. Okay. Some of what makes this population, some of what makes you vary in height is the fact that you inherited different genes from your parents. Some of what makes you different in height is the fact that you grew up in different environments as children. This is an attempt to kind of partition out the total variance into this population into a component that is due to genes and a component that's due to environmental variation. Now, you have to be really careful with this because you hear it used a lot in ways that are misleading. You know, somebody might tell you that your height is 70% genetic and 30% environmental. That does not, emphatically not, mean that you can say, okay, I am 70 inches tall. I got 49 of those inches from my parents, and the other 21 of those images by eating lots of Wheaties when I was a kid. Right? Your own personal height is just one number. It doesn't have a variance, right? What's the average of 3? Right? It's got no calculated variance at all. And your personal height is 100% genetic and 100% environmental. That's not very helpful, but you can't distinguish the difference between genetic and environmental effects on a sample size of 1. Your own height is just what it is, and it doesn't have a variance. Variance is not the fraction of an individual's trait that is down to genes versus down to the environment. It's the fraction of variance over an entire population that is down to genes versus down to environment. Okay, think I'm explaining this possibly well, although some of you are probably looking at the screen like I've just swallowed a live weasel or something. You can think of it like this. V sub G would be the variance in a population if all of its members had an absolutely identical environment. We could measure VG by taking a population of a species of plant and growing them all in one greenhouse and giving them exactly the same amounts of water and light. So if we took a population and raised all members in an identical environment, the environment wouldn't vary, and all members of that population would have the same environment. So anything that made them different could only be their genes. So that would be V sub G, variance in a population that's due to genetic variance. And then we could measure VE by taking a bunch of plants that were all clones, maybe cuttings from each other, and growing them in a range of different environments. So if we had a population of plants that were all genetically identical, but raised in different environments, the plants might vary in their height, let's say, and that variance would be entirely V sub E, variance due only to environmental effects because there's no genetic variability at all. And then in a natural population, the total phenotypic variance would be the sum of those two. The total phenotypic variance would be the sum of the variance due to genetic variation and the sum of and the variance due to environmental differences. Okay, I hope I'm making that not entirely unclear. I'm flailing because I'm looking for this. I usually diagram this on a chalkboard, but I hope I'm not being completely obscure. Like I said, these equations only apply to variance measured over a population. It's not an individual trait, right? My height, let me see, if I don't slouch, I probably top out at about 74 inches, or at least I did in my younger years. Okay, the variance of my height is, okay, my height is 74. The average height of Dr. Wagoner is also 74, right? It's the average of a single number. The difference between my height and the average height of all people named Dr. Wagoner is 0. And so the variance is 0. I have no variance in my height because I'm a population of 1. So there's no possible way I could say that 70% of my 6'2" height is due to having a tall dad, And 30% of my 6'2" height is due to, actually, I mostly ate Cheerios as a kid, not Wheaties. Either way, it doesn't mean anything because the equations only apply to variance within a population. Variance is not a feature that individuals have at all. It's only calculated relative to a population. And as we'll see, the equations do not necessarily apply to variance between different populations. You can do the same kinds of studies on different populations and get different answers as to how much variance is genetic and how much is environmental. Okay, we can take something called broad sense heritability, and it's usually represented by capital H squared. And that is simply the genetic variance divided by the total variance in the population. We call that heritability, but you have to be really careful because you can have a trait that is genetically inherited but has no genetic variance. Look down below. We do know that there are a number of genes that are involved in the development of the nose, right? Okay, it is because of your genes that you have a nose. But if I was measuring the number of noses per person, everybody has exactly one nose per person, right? Nobody's got any extras like maybe growing out of the back of your neck or anything like that. At least I hope not. So if I was to measure the number of noses in this student population, right? Each one of you has one nose. The average number of noses per population is 1. We calculate variance as the sum of the squares of the differences, but the difference between the number of noses each of you has and the average number of noses is 0, right? 1 nose per person. And the upshot of that is that if we try to calculate the broad sense heritability of the number of noses per person, we get 0 divided by 0. So even though we know that genes have something to do with noses, it does not have definable heritability because the trait doesn't have any variance at all. You could even have a trait that is inherited genetically and has 0 heritability in a population if it has no genetic variance. Imagine some population of humans where everybody has identical skin color. Some of those people go to tanning salons and some of those people are goths and stay out of the light. We'd find a range of skin colors in that population. Sorry, I just saw a couple of things in the chat, just checking on those. We'd find a range of skin colors in that population, but within that population, you would have 0 heritability because genetic variance would be 0. So if you have a trait that varies in a population only because the environment varies, the trait may be inherited genetically, but still have 0 heritability. Okay, that may seem a little bit counterintuitive, but heritability is a different concept from simply being inherited. I can probably show you a slightly better example of this once we've plowed through a couple more things. We can define the narrow sense heritability of a trait to be the additive genetic variance as a fraction of the total phenotypic variance. Additive genetic variance is the amount of variance that depends only on the number of alleles. You've just seen, or you're about to see, a great example of this because I had you tracking down cats in shelters that have white spotting, right? remember that this is a trait that shows in white spotting is governed by a single gene with two alleles that show incomplete dominance. So a cat that has no copies of the spotting gene won't have any white spots. A cat that has one copy of the spotting gene will have relatively small amounts of white, maybe just white feet or a white star or a white lower face, kind of like Batman or something. A cat with two copies of the spotting gene will have so many white spots that it will be a mostly white cat with only smaller areas of colored fur, maybe just white, maybe just colored ears and a colored tail, maybe a colored stripe down the back, but the rest of the cat's going to be white. So if you have no copies of the spotted gene, you don't have white spots. If you have one copy, you have a few white spots. If you have two copies, you have so many white spots that you're mostly white. That's an example of additive genetic variance because it depends on the number of alleles that you've got. You can have 0, 1, or 2 alleles for white spotting, and that's correlated with none, a little, or a lot amount of white spotting. So the genetic variance is entirely additive because what is making your cats look different from each other is the number of alleles that they've got. The problem is that in some cases, what is making your cats look different from each other is not just the number of alleles that they've got, it's things like genetic dominance. For example, if memory serves, long hair is dominant to short hair in cats, but this is actually a fully dominant trait. So you can't tell the difference between homozygous dominant and heterozygous cats. Some of what is making your cats look different from each other in fur length is additive, in that if you have no long-haired alleles, you'll have short fur, and if you have some long-haired alleles, you'll have long fur. But some of what is making your cats look different from each other is the effects due to genetic dominance. And we can actually partition genetic variance into the additive variance, which is the amount of genetic variance that's due to the number of alleles, the dominance variance, which is the variance due to dominance effects among genes, and interaction genetic variance, which is variance due to epistasis, variance due to interactions between genes. A kitty cat example would be things like, the tabby pattern is one. You actually have two separate genes, each with different alleles, and all cats carry a tabby gene, but they don't express it unless they're also carrying a second gene. So if we looked at variance in the degree of tabby striping in cats, some of that would be due to the number of alleles for the different tabby patterns that a cat has. Some of that would be due to genetic dominance, because some tabby patterns are dominant to others. And some of that would be due to genetic interaction, because cats don't show tabby color unless they've got the right loci at a different locus called the agouti locus. So what makes your cats all look different is partly due to their genes, and then that genetic variance can be partitioned into variation due to additive effects, variation due to dominance, and variance due to interaction. And I tell you what, I'm going to stop there, and we'll pick this up with a nice example. This is the scarlet tiger moth. Used to be Panaxia dominula. Now they call it Callimorpha. It's got spots on the forewing, And what I want you to see, you can see those, and that number of spots happens to be governed by a single gene that has two incompletely dominant alleles. So the top one has a genotype we can write as big S, big S. The bottom one is homozygous recessive, little s, little s. The one in the middle is heterozygous, and you see it's got an intermediate amount of spotting on those front wings. And what I'm going to do on Thursday is introduce how we can take a population and use it to actually calculate narrow sense heritability. By the way, I'm going to give you one little homework assignment. And I'd like you to turn this in this coming Thursday. Actually, I tell you what, email this to me before lecture. Try to do it by tomorrow night. Here's what I want you to do. Send me your own height in inches, right? So if you're 6 feet tall, that is, of course, 72 inches. No, don't even do that. Send me the average height of your biological parents, if you know it. If you know how tall your biological mom and dad are, calculate their height in inches and take the mean of that, average those two numbers, and record that as mean parental height. Then the second thing is take the height of you and the height of any full siblings that you've got. Leave out half sibs out of this. Leave that out. Leave out half brothers and sisters if you have them. Calculate their mean height in inches and record that as mean offspring height. If you cannot do this for any reason, you don't have to. I'm not awarding points for it. It would be an interesting way that I can, that we can participate and I can make these concepts a little bit more concrete. But if you, for any reason, if you're estranged from your bio parents, if you don't know who they are, maybe some of you were adopted or fostered, you don't have to participate in this if you don't want to. It's not mandatory. If you can, send me the mean height of your parents and the mean height of all of their kids. If you're adopted, you can ignore this. But if you can, and if you remember to, some people are just going to forget, and it's not a big deal, but try to find out the mean height of you and your full siblings and the mean height of both parents. Send me those numbers by evening tomorrow, and I'll show you what we can do with them to calculate narrow sense heritability. So I will now stop the recording.